Hi, my name is Jonathan Flintoff and I'm a first year PhD student at the Queensland Brain Institute under the supervision of Associate Professor Tom Byrne. So my project today that I'm going to pitch will be establishing a novel task to investigate cognitive impairment in neuropsychiatric disease. So cognitive impairment is a feature of various different neuropsychiatric diseases such as schizophrenia, depression, bipolar disorder, OCD, as well as many others. So some of the cognitive areas that are affected in these neuropsychiatric illnesses are attention, memory, social cognition, processing speed, as well as executive function. So our lab primarily focuses on schizophrenia, and it can be divided into three primary clusters of symptoms. So some of these include the positive symptoms, which are things like hallucinations and delusions, negative symptoms, which includes anhedonia, which is an inability to experience pleasure. But you also have cognitive symptoms. And so these cognitive deficits are major factors in the functional disability within schizophrenia. So current medications target the positive symptoms, but they do not target any of the negative or cognitive symptoms within schizophrenia. So this is why in my project I wanted to work on improving the translational tests that we have to address the lack of progress being made in developing treatments for this area. So within behavioral flexibility, this is part of executive functioning. So an example of this in humans might be a task where you have to select a correct stimuli or an incorrect stimuli. So as you don't know the rule beforehand, you select a stimuli and you get feedback. Once you learn the correct stimuli, you have to be able to suppress an action of selecting the incorrect stimuli, acquire the correct stimuli to make the right response, and you have to be able to maintain this response. So behavioral flexibility can be summed up as the ability to dynamically adapt within an environment. So in schizophrenia, this behavioral flexibility is impaired and people are unable to adapt to different changes. So within the preclinical models that we currently have of rodent assays, we have simple discrimination tests where we have a rule and we reverse it within rodents. Some of the later ones that have been developed use touchscreen technology, and this provides higher translational validity, but the stimuli used are still quite different from the ones we use with humans, lowering the translational value of the tests. Some of the more complex ones are the extra-dimensional digging tasks. So these tasks are quite manual and slow to put a high number of animals through. So this provides a kind of bottleneck in the therapeutic development of treatments, as they're quite slow. So that's why in my project I wanted to develop an automated operant test of flexibility. So we already have the chambers that have been set up, and so this consists of an operant chamber where we have a central screen, we have a nose poke, and we have two reward receptacles. So in the simple discrimination task, the animal has to learn an association between an image and a correct response, which may be making a head entry into the right reward receptacle. So this is a basic measure of flexibility when you have the right reward receptacle providing a correct response and the left providing an incorrect response. So when we show different images, we want to teach the rodent to associate a new rule of image and reward pairing. So once we've developed this simple discrimination task, we want to be able to reduce the training time it takes for these animals to learn this reward pair association, and we want to investigate which stimuli facilitates the learning of these reward stimulus pairs. So we also want to look at automating the rule changes. So once an animal selects the correct rule a certain number of times, then the rule changes. So this rule change might be having to select shapes of a different color, number, shape, or select image stimuli based on the time that they're presented. So we want to be able to increase the complexity of the stimuli being shown while also reducing the training time. So the primary outcome measures that we're going to be recording on this task are the number of rule changes learnt. We also want to record the perseverative errors. So these are the errors in which the rodent is presented with a new rule. This might be a new image, a number of images, or color yet they continue to make the wrong response. So we also want to record the random errors that the rodent might make. These are the incidental errors that occur over the period of a session. So we also want to record the session time as well, and this will give us a measure of how long it's taking each rodent to complete the reversal learning and be able to adapt to these new rules. So to validate this new task, we want to be able to do a lesion experiment as well. So in humans, behavioral flexibility is controlled by the prefrontal cortex, and the homologous region in rodents is the medial prefrontal cortex. So we want to be able to lesion this part of the brain and then retest the animal on the task to see whether those measures of rule changes learnt decrease, perseverative errors increase, number of total errors increases, as well as session time decreases. So if we see these measures begin to decrease, then we know that this task is a medial prefrontal cortex dependent task. So once the task is set up, collaborations will be desirable to validate this in a preclinical model of schizophrenia. So this might include a developmental model such as vitamin D deficiency, which we do in our lab. We may also want to look at a pharmacological model such as ketamine. These pharmacological models also are able to recapitulate some of the cognitive impairments we see with patients in schizophrenia as well. We may also want to look at a genetic knockout model, and this might be something like the DISC-1 model to look at cognitive impairment. And so using these preclinical models, we may be able to look for biomarkers of cognitive impairment as well. And this might take the form of metabolic dysfunction. So we may want to look at glucose regulation in the medial prefrontal cortex of these animals to see whether there's a dysregulation there that predicts cognitive impairment. So why is this research important? The point of this task will be to bridge the translational gap between the animal tasks and the human tasks we currently have. So if we have rodent tasks that are highly translational and similar to the human tasks, we have more valid predictions that we can make about pharmaceutical interventions. So this is going to help us develop novel therapies to be able to target the cognitive impairment we see in schizophrenia. So as well, we want to be able to automate these tasks so that they're higher throughput, so that different labs are able to put a high number of animals through these tasks and be able to get results quicker. Finally, we want to look at the search for a biomarker that predicts cognitive impairment. If we can find a marker of metabolic dysfunction that may be able to predict cognitive impairment, we may have some hints as to the therapeutic strategies that might work in reducing cognitive impairment in patients. So I think it's important to remember that there will probably never be a perfect model of cognitive impairment in neuropsychiatric disease, but it's essential that we continue to develop tests that are more sensitive and specific to specific areas of cognitive impairment. So that way we can target treatments and we're able to improve the lives of people living with neuropsychiatric disorders in their day-to-day life. Thanks.